Welcome, everybody. As everybody's filtering back in, I do want to start by giving a shout out to the McKernan brothers, Brendan, Brian, and Kevin. They are the prime movers of CannMed, and they are an amazing trio of brothers, an amazing group of entrepreneurial intellectuals, and it's been great to get to know them the last few years and to work with them, my family and theirs. We've become good friends and collaborators, and I appreciate their invitation for me to share some of our work at Project CBD and my focus lately on psychedelics. You know, yesterday you heard a lot about cannabinoid acids. So today you will hear about acid and cannabinoids. You know, there is sort of a revival of scientific inquiry now into psychedelic therapy and its potential there. What I want to focus on in this discussion is how the endocannabinoid system actually mediates many of the key aspects of the psychedelic experience. I won't ask for a show of hands of who's familiar with this personally. I don't think I could see the hands anyway because of the light. But so buckle up. It's an interesting discussion. So let's start with the classical psychedelics. In the early 1950s, really the classical psychedelics were grouped into three categories. You have the tryptamines. That's DMT is the active hallucinogenic component. You know, that's part of ayahuasca, iboga, rue. There's the phenethylamines, that's mescaline, peyote, and what I'm calling the lysergamines, LSD, which is interesting. LSD is, or lysergic acid could be derived both from a fungus and from plants, the rye fungus, and also from the morning glory seeds and the Hawaiian baby woodrose. What's important about the early psychedelics is how they catalyzed the field of serotonin neuroscience. In 1954, there were two scientists from the Rockefeller Institute, Woolsey and Shaw, that drew attention to the structural similarity of the LSD molecule and the serotonin molecule. And, you know, at that time, this was really the beginning of the whole discussion and the lingo that we're used to these days of neurotransmitters and receptors. That's really where it all began, in the context of serotonin neuroscience, of which LSD played such a catalytic role. So the early LSD research, and LSD was really the star of the show in the 1950s, or at least up until the very late 50s. That's really where the focus was in terms of medical science research, and there's quite a bit of it. There were really two paradigms, if you will, of looking at LSD. Initially, it was really looked at as a psychotomimetic agent, something that would mimic madness, that would create an artificial schizophrenia. This gave rise to some very interesting theories and possibilities that if you could find something that could be an antidote to the LSD schizophrenia, maybe it would work for real schizophrenia and so forth. But as the 50s proceeded, that way of looking at LSD and psychedelics was supplanted by what you could call the psychedelic paradigm, if you will. And it was Dr. Humphrey Osmond who coined the word psychedelic, and it simply means mind manifesting. It's a very neutral term. It doesn't have any associations initially with the 1960s and Paisleys and Dayglow and so forth. It was an appropriate term. All it meant was that these drugs were psychodynamic and had a way of bringing up, making conscious whatever is unconscious in almost a value-neutral way. And that's why it was useful for therapy, it was seen. So in terms of the therapeutic indications, initially what doctors got onto in the early 50s was the potential for LSD as a treatment for alcoholism. And it was Dr. Humphrey Osmond who also was involved in that research very early on. And it was very exciting. They had very good results that they couldn't really explain. But it was so substance abuse, depression, and trauma were really the three areas that seemed to be indications that LSD was potentially useful for in a therapeutic context. So you had, from the 1950s through the 1960s, you had more than 1,000 clinical papers about LSD and psychedelics. And they described 40,000 patients. There were several international conferences about LSD therapy. Some of these were actually sponsored by the Central Intelligence Agency in the United States. Overall, there was a lack of adverse reactions reported to LSD when it was in a controlled clinical setting. And a meta-analysis that was done in, I guess, 2016, looking back on this research, the meta-analysis of the control trial said that, that there was a demonstration of a consistent and clinically significant beneficial effect of high-dose LSD therapy. So what happened was, because of the 1960s and the association of LSD with the political turmoil during those years, all the psychedelic research was basically put on hold and stopped. And there was a hiatus. That was really basically a political reaction to the non-medical use of LSD in the 1960s. And for 2 decades, really nothing was happening in terms, at least in the United States, in terms of research into psychedelics and the therapeutic potential. And it started up again in 1992, but instead of the focus being on LSD, that was supplanted by scientific research into psilocybin, the magic mushroom extract, if you will. And so there really wasn't much work on LSD since really the mid-1960s, but the work on psilocybin proved to be very, very interesting. There were 2 independent randomized controlled trials, and randomized controlled trials for psychedelics are obviously sort of challenging because it's very difficult to hide from someone the fact that they've been given a drug if they're tripping out, But nonetheless, there's been efforts made in this area, and there were 2 very interesting independent randomized controlled trials that showed psilocybin had significant long-lasting efficacy against cancer-related anxiety and depression. So it wasn't like you take the dose and you feel better while you're under the influence of the drug, that effect of the antidepressant effect, the anti-anxiety effect, lasted. There was a 2011 study at Johns Hopkins that reported that psychedelics can trigger mystical quantum leap type of experiences in healthy people, resulting in, again, long-lasting, sustained positive personality changes. A large percentage of people in these studies rated the psilocybin experience as one of the most meaningful of their lives. And that's a sort of you hear that again and again in this context. Just to look a little bit closer at some of the interesting work done with psilocybin before we moved into the endocannabinoid system, there was this study from Imperial College. You see the abstract is posted, where they actually used fMRI to analyze brain activity, brain frequency under the influence of psilocybin. So this is your brain on drugs. You see some of the images there. So the study from Imperial College was psilocybin for treatment-resistant depression and MRI. Looking at the MR brainwaves. So this is at Imperial College. They probed the neural correlates of psychedelic experience, and they looked at the changes in brainwave activity, changes in brain activity under the influence of psilocybin. This is the first time this was seen, this kind of imagery for psychedelic experience. A drawback of the study, there was no placebo control for this. It was a relatively small number of patients, but for all 19 patients, the psilocybin experience was shown to be efficacious with assisted psychotherapy along with it. There was always a talking session that went with the therapy before and after, and that was important, that was part of it. And so for treatment-resistant depression, for all 19 patients of breakthrough experiences, and what's the reason for this? Why does this happen? The analysis given by the scientists from Imperial College, they felt that what they saw with the imagery had to do with something called the default mode network of the brain, which is not so much a brain area as a bunch of areas and a circuitry between the areas. And it's almost like that's what's going on when you're on automatic pilot. It's said to be the neurological basis for the self. That might be an overstatement, I think. But what they found was that under the influence of psilocybin, the default mode network was basically suspended, and something else was going on. It was a great deal of activity when ordinarily there wouldn't be. So there was a follow-up study or follow-up analysis of all the patients 6 months later, and again, they found the antidepressant effects lasted all this time from, well, actually, they used ultimately 2 doses in this. So you have this long-lasting effect. As a result of this study and others, the FDA has actually fast-tracked psilocybin as a treatment for treatment-resistant depression, which is quite exciting. That's in progress, and we might see psilocybin approved, although there's some caveats there with who would get the approval and so forth. So that's what they saw, and that's what they've seen in other studies. It's not like there's been extensive research with psilocybin since 1992, and it started again, but there's enough to really, raise a great deal of interest and hope. So I want to switch gears a little bit and talk about ketamine, another hallucinogen, at least in high doses. It acts as a psychedelic. Ketamine already is widely used as an antidepressant off-label. Ketamine is an FDA-approved drug, but not for depression. It's approved as a dissociative anesthetic, as a painkiller. And there's been a couple of papers published within the last 2 years that show that the pain-killing effects of ketamine, both in terms of central and peripheral based pain, are both mediated through the endocannabinoid system. And I think that there's enough compelling evidence to show that the antidepressant effect from ketamine is also mediated by the endocannabinoid system. We'll go into that for a moment. What's interesting about the treatment centers that have popped up all over the place now that people can get a ketamine treatment, again, they're finding that the effect of the antidepressant effects last more than just the drug itself. They will last for a week or 2. And for people who are suffering from refractory depression, this is a godsend. The other thing is that they found in the research that the endocannabinoids actually will potentiate the effect of ketamine. The way this is working, and this is, actually, I'll need my glasses for this one, but you could see the title of the abstract. It's focusing on how the interplay between cannabinoids and the NMDA glutamate receptor. That glutamate receptor is actually an ion channel, and what ketamine does is it blocks this ion channel, and as a result of doing that, it triggers the release of something called brain-derived neurotrophic factor. And BDNF has been described as fertilizer for the brain, and what it does, it stimulates neurogenesis, the creation of new brain cells in particular areas of the brain, which happens into adulthood, actually, this process. But what ketamine does, it sort of boosts this effect. And not just for neurogenesis, but neuroplasticity and synaptogenesis, actually the growth of new dendrites on a cell. So ketamine's rapid antidepressant effect involve enhanced BDNF-induced neurogenesis and synaptogenesis. There was one study that showed a 10 milligram per kilogram dose of ketamine produced a robust increase in dendritic spine density, meaning the growth of the dendrites on the cell, in the prefrontal cortex of rats. Okay, so looking at this a little bit closer, BDNF and the endocannabinoid system. Unfortunately, I cannot read very well because but on the cellular level, the cannabinoid system regulates the expression of brain-derived neurotrophic factor on a cellular level. And that's, so what we have going on here is ketamine, which is stimulating a brain-derived neurotrophic factor, and that process is regulated by the endocannabinoid system. That's what this paper shows. To explore this a little more, let's look at the endocannabinoid system in neurogenesis in general. What we know from various research is that the endocannabinoid signaling actually choreographs the creation of the nervous system and the brain in the embryo. So it's involved in regulating the process of creating brain cells and neurons, literally. And that process, while it drops off significantly after birth, it does continue. And there are two what's called neurogenic niches in the brain, in the hippocampus. There's the subgranular zone of the dentate gyrus and the subventricular zone of the lateral ventricles. These are the two areas in the hippocampus that have identified as the areas where neurons are created. And these are areas of the brain that are heavily populated by cannabinoid receptors. And it's been shown that an activation of the CB1 receptor in the brain actually stimulates neurogenesis. And it's also shown that CB2 receptors in the brain, and there's some controversy if they're generally in the brain, CB2, if they're only emerge when you have an injury or when you need it to be there, but that the CB2 receptor, the CB2 cannabinoid receptor, drives neurogenesis and improves functional outcome after a stroke. It's also been shown that a pharmacological blockage, if they give a drug that will block the cannabinoid receptors from acting, or if they create mice that they have the cannabinoid receptors deleted, that results in impaired neurogenesis. Chronic stress depletes neurogenesis. Pha, which is the enzyme that breaks down cannabinoid, if you inhibit Pha, it results in an increase in neurogenesis. So they've determined this in various ways, what's going on, the scientists. Neurogenic activity is most robust, at least after the child is born, during childhood. It tends to decline over age, and it can decline to the point of no activity in an elderly person. And the gradual reduction in new brain cells creation has been linked to cognitive decline and neurodegenerative disease. Exercise, enriched environment, mental engagement, intermittent fasting, and a good night's sleep, all those boost neurogenesis. So again, this is a process that's continually happening in our brains to one degree or another, but there are certain things that will boost that process, and it turns out that psychedelic drugs do as well, as I'll show in a moment. So neurogenesis in particular areas of the hippocampus, but neuroplasticity throughout much of the brain. The endocannabinoid system regulates neuroplasticity throughout the brain, including the striatum, where various forms of bidirectional endocannabinoid-mediated plasticity occur. Again, this is what the science is showing. So endocannabinoid-mediated synaptic plasticity within the hippocampus is well documented. Synaptic plasticity, basically what that means is changes in the strength between the neurons themselves. Long-term potentiation involves strengthening connections between neurons. Long-term depression in the hippocampus basically means weakening those connections, or that's how we literally clear memory in the hippocampus through a process known as long-term depression. We don't need to remember everything. Things that are important now, we might, gradually those. As Meshulam says, nature is stingy. If you don't need it, we don't use it. If we're not using those memories or relying on them, they fade away as part of this process. So endocannabinoid signaling facilitates adaptive learning, the brain's ability to make new neural connections and correct abnormalities from chronic stress. And it's the CB1 receptors, the cannabinoid receptors in the brain that mediate changes in synaptic plasticity. It's been shown that the CB1 knockout mice have reduced dendritic activity and within the prefrontal cortex. Again, it all ends up saying the same thing. This, looking at substance abuse, because recall now with LSD and the early research, substance abuse, alcoholism was something that showed a great deal of potential in terms of what the psychedelics maybe could do. And there's been interesting work on the endocannabinoid system in substance abuse. This is one study from 2011, endocannabinoid-mediated synaptic plasticity and addiction-related behavior. To look at this a little more closely in terms of alcoholism and neurogenesis, what we've learned is that substance abuse causes a disruption in the synaptic plasticity of the brain circuits involved in addiction, with the alteration of normal endocannabinoid activity playing a very important role. That chronic alcohol exposure reduces endocannabinoid activity and disrupts adult neurogenesis. And the endocannabinoid system is one of the most relevant biochemical systems mediating alcohol addiction. Adult hippocampal neurogenesis is a key factor involved in drug abuse, and it may provide a new strategy for the treatment of alcohol addiction and dependence. In terms of depression, neuronal atrophy is related to depression. Plasticity, you can think of it as plasticity is a sign of health. Dendritic atrophy is a sign of a waning health. A loss of dendritic spines in the prefrontal cortex is a hallmark of depression and neuropsychiatric disease. Neuronal atrophy is a contributing factor to various mood disorders, anxiety disorders. The behavioral effects of some of the standard antidepressants are contingent on enhanced hippocampal neurogenesis, and they work by promoting neurogenesis. And facilitation of neurogenesis could be considered a treatment strategy for clinical depression and for various neurological diseases like Alzheimer's, MS, Parkinson's, and so forth. Turns out THC promotes neurogenesis. THC enhances neurogenesis in the hippocampus, improving cognitive function of rats. THC stimulates the creation of new brain cells by activating CB1 receptors. THC administration promotes the upregulation of BDNF gene expression, and THC enhanced the markers involved in all stages of the neurogenesis mechanism. And as I think it was Dustin pointed out earlier today, ultra-low doses of THC have actually been proposed as a possible remedy for brain injuries and age-related brain decline. Ultra-low doses. CBD is also neurogenic. CBD induces a rapid and sustained antidepressant effect through increased BDNF signaling and synaptogenesis in the prefrontal cortex. The pro-neurogenic effects of CBD might explain some of the positive therapeutic features of CBD-based compounds. CBD attenuates the decrease in hippocampal neurogenesis and dendrite spine density induced by chronic stress. This is interesting. Low doses of CBD increase cell proliferation and neurogenesis, while higher doses decrease this. And go back again to what people have been talking about at this conference in terms of dosing, that oftentimes less is more, and that would seem to be the case in terms of CBD and neurogenesis. So here we have a paper that came out last year by scientists from the University of California, Davis. And you can read the title of the abstract. Psychedelics promote structural and functional neuroplasticity. So there we have it. To go into this a little bit more, psychedelics not just promoting neuroplasticity, but neurogenesis. That's part of the way they work, and that's part of how they confer an antidepressant effect. So classical psychedelics upregulate BDNF and promote enhanced neurogenesis and synaptic plasticity, even more so than ketamine. Psychedelics promote changes in the neural structure and dendritic growth across vertebrate and invertebrate species, and that means it's through an evolutionary conserved mechanism, which is something that we've needed, for millions of years. It's something that's very important, and that's why it's been conserved. The positive impact of psychedelics in terms of substance abuse and depression is contingent on psychedelic-induced neurogenesis and neuroplasticity. But it turns out there's several species of non-psychedelic mushrooms that also promote enhanced neurogenesis and neuroplasticity, like turkey's tail, lion's mane, cordyceps, things that we're learning about through Paul Stamets and others, and that we can access legally. I want to focus for a moment now on the 5-HT2A. This is the serotonin 2A receptor, because that's the receptor that in 1998, France, Vollenfeder, Swiss scientist, he showed that LSD and psilocybin, they exert their potent hallucinogenic effects through this receptor. So the serotonin 2A is the psychedelic receptor, and it is responsible when it's activated for many of the signature effects we associated with psychedelic drugs. So what are these signature effects? Well, vivid, kaleidoscopic visions for starters, but that's only the beginning. Radically altered sense of time experience, experiential density, dazzling immediacy. This is what we associated with the psychedelic experience. And this is all being mediated by the 5-HT2A receptor. It's responsible for these effects. And it's also responsible, interestingly, for changes in gene expression in the cerebral cortex and changes in synaptic plasticity. It's been linked to this receptor mediates it. The higher 5-HT2A binding affinity, like LSD has a very strong binding affinity, that predicts a more potent neurogenic and neuroplastic effect, the greater the binding affinity for the ligand. 5-HT2A receptor antagonists block the ability of psychedelics to promote neurogenesis, synaptogenesis, and synaptic plasticity. So it's again a confirmation this is the pathway that the psychedelics are conferring this effect. Aberrant serotonin 2A signaling is linked to headaches, mood disorders, hallucinations. These receptors are dysregulated in the prefrontal cortex of schizophrenics. Interestingly, CBD also binds directly to this receptor. So LSD and CBD bind directly to this receptor. CBD is considered more active at the 5-HT1A receptor. That's typically what we've been hearing about at this conference. It's a receptor that mediates the anti-anxiety effects of CBD, perhaps the antidepressant effects, I'm not so sure, but it's one of the main receptor partners for CBD. CBD does not bind well with the cannabinoid receptors, but it binds very powerfully with serotonin 1A, less so with 2A. But it does bind to it directly. And it turns out that the serotonin 2A receptor is more prevalent in the brain and body than 1A. It's expressed in nearly every cell type, and its highest density is in the neocortex. So CBD, it exhibits enough affinity at this receptor to talk about it in the context of this is a meaningful pathway for CBD, and it's not something that's generally discussed much. The focus usually is in terms of 1A. But the question is, how is it possible that you have LSD, which is the most psychedelic drug you can imagine, and CBD is about the most unpsychedelic drug you can imagine? How is it that they're both binding to the same receptor and having such different effects, and that obviously CBD isn't making a person trip out? And I think this is through something called functional selectivity. And you could see it, I wish I had the image up on the screen, but if you have sort of a highway and you're hitting this receptor, and then with LSD and CBD on either lane, and then it separates. But then it connects later downstream because both of them are promoting neurogenesis and neuroplasticity, but one is intensely psychedelic and one is not. So functional selectivity, what does that mean? That different ligands are the key that's fitting into the locks. So you have the different ligands, CBD and LSD, signaling through the same receptor, in this case 5-HT2A, can produce distinct physiological responses that culminate in similar downstream effects, in this case, the downstream effects, again, neurogenesis and neuroplasticity. So now THC does not bind directly to any of the serotonin receptors, unlike CBD, but THC does engage in what scientists refer to as crosstalk between between the endocannabinoid system and the serotonin system. And it does this through something called receptor dimerization. And what that means, when two different receptors, they kind of get entangled and they conjoin. And it's increasingly understood that this happens a lot, but it's not clear why it happens or exactly what it means. But the preclinical research, interestingly, shows that that the CB1 cannabinoid receptor and the 5-HT2A serotonin receptor, when they form what's called a heterodimer, that this is responsible for mediating both THC's pain-killing effects as well as its memory impairment effects. And that the CB1 5-HT2A receptor heterodimers are expressed and functionally active in specific brain areas involved in memory impairment. Interestingly enough, there's another heterodimer between CB1 and adenosine, A2A, that when CBD activates that complex, it restores memory. In other words, it counteracts THC's memory impairing effects. So what about cannabis now? Can you consider cannabis to be a psychedelic? I think it's really a matter of dose, because small doses, moderate doses are relaxing for most people, fun, whatever. But high doses can be psychedelic. And what I wonder is this heterodimer between the CB1 receptor, which THC will activate, because it's linked now to 5-HT2A, is that responsible for the hallucinogenic effects of very high doses of THC? There's been some interesting research, of all places, from the University of Kansas. They did a series of studies where they showed that cannabinoid receptor agonists upregulate and enhance 5-HT2A receptor activity. Now, they weren't working with THC, they were working with synthetic agonists, but the implication is, possibly, that cannabis will actually a high dose will upregulate the serotonin 2A receptor. So you have another connection there between them. Fitzhugh Ludlow was the first American writer to write about hashish. This is from before the Civil War. He has this wonderful phrase about how hashish loosens the girders of the soul. As I think that's maybe like the default mode network being suppressed or suspended. But this kind of experience of a high dose of cannabis or cannabinoids being hallucinogenic, like mushrooms, there was actually a study done in 1969 by Dr. Harris Isbell, who happened to be a key player in the CIA's work with LSD, comparing high doses of pure THC to LSD. And he concluded that high doses of THC are hallucinogenic. And I don't know that there's been any other formal study other than this one going in 1969. So it's my speculation that this conjugate between the cannabinoid receptor and the serotonin receptor, this may be implicated in the neurobiological underpinnings of hallucinations caused by high-dose THC. Teasing this out a little bit further, this is a really fascinating study by Dave Nichols and his group at Purdue from 2006. And you read the title of the abstract, what it's saying is that when you activate the serotonin 2A receptor, it results in the formation and release of endocannabinoids. So again, you have this connection between the serotonin system and the endogenous cannabinoid system. So CBD, LSD, psilocybin, activating 5-HT2A will induce the creation, the formation, the release of endogenous cannabinoids, and in particular 2AG is the cannabinoid that's released. There was a follow-up study 3 years later, basically affirming the same thing, that the serotonin invokes endocannabinoid release, that results in activating the cannabinoid receptors that function in this retrograde, that function in a way to sort of turn down a neurotransmission, or like a dimmer switch almost, of other neurotransmitters. But what I found striking in this study, it stated that based on the extensive distribution of CB2 receptors and CB1 receptors, it seems likely that this mechanism could mediate many of the actions of 5-HT2 receptors throughout the body. And this could be it could be a possible answer to the puzzling phenomena of how CBD seems to be able to exert effects that are very similar to the effects of the cannabinoid receptors being activated. It's not understood how that works because CBD doesn't really directly activate them. So it might be through this channel. It might be through so what we see really going on here, okay, we have an agonist like LSD or CBD binding to the serotonin 2A receptor. This releases endocannabinoids, 2AG, which then bind to cannabinoid receptors, CB1 receptor, which promote neurogenesis. That seems to be the four-step pathway here. So this is then the last slide. Interaction between 5-HT2A receptors and endocannabinoid system is fundamental to the neurogenic and antidepressant effects of classical psychedelic drugs. 5-HT2A-induced 2AG release promotes neurogenesis, neuroplasticity, and new neural connections, and that seems to lead people to new perspectives on life somehow. There's a phrase in neuroscience, neurons that fire together, wire together. The brain kind of can get stuck in certain habits, and it seems that these psychedelic drugs can sort of catapult people out of the ruts, the brain ruts that they're in, but with a very long-lasting effect. And that's what's so puzzling. Why do these things have such a long-lasting effect? And I don't think science really has an answer to that. I think you probably want to look at many different fields to perhaps get an answer, because this is an existential thing we're talking about, a major life change that results from these experiences. And I would look at certain philosophers like the phenomenologists like Merleau-Ponty, who the Phenomenology of Perception, published in 1945, actually talks about mescaline very early on. And what he says, interestingly, is that one of the key features of mescaline is synesthesia, that you're seeing sounds, hearing colors, that kind of thing. It's another signature effect. And what he concludes in this massive philosophical tome, and he, by the way, was the chair of the Sorbonne philosophy department, and he was a leading philosopher in France. He said that basically synesthesia, when you really analyze it, is the normal way of perceiving that gets sort of overlaid by conditioning as we go through life, and that the psychedelics sort of remind us of this experience. So I'll leave it at that. Take some questions if you'd like. Thank you.